Hi, everyone. My name is Nicole White, and I am the Executive Director of Rare New England. And welcome to the Medical Genetics Career Fair. Rare New England's mission is to bring together New England patients, families, and providers touched by rare and complex disorders. We offer educational opportunities, create awareness of available resources, and build foundations for support to improve patient quality of life and support the professional needs of healthcare providers. Here tonight, you will see our host, Dr. Amy Kritzer. We have our guest speaker, Dr. Jacqueline Amadorian, Dr. Stephanie Sakharov, and Dr. Monkolek. And we'd just like to thank our sponsors for helping us, with the support. We wouldn't be able to provide this great education. So thank you to Ultragenyx, Vertex, Horizon, and Homology for your support, as well as the New England Regional Genetics Network as well. And I am going to pass it on to our host for this evening, Dr. Amy Kritzer. Dr. Kritzer is an attending in the Division of Genetics and Genomics at Boston Children's Hospital.
Thank you so much, Nicole. And I'd like to welcome everyone to our career panel tonight. You're going to have an opportunity to hear from some fabulous team of people with a diverse interest in genetics. Each one of them will present on their area of specialty, and then we'll have an opportunity to take questions and answer questions that have been previously submitted from our panelists. I'll introduce myself. I'm Dr. Amy Kritzer. I'm the clinical director of the Division of Genetics and Genomics at Boston Children's Hospital. I am a clinical geneticist and a biochemical geneticist. I spend most of my time caring for children and adults with rare inborn errors of metabolism, enzymes deficiencies, and work with a team of genetic counselors, dietitians, students, genetic counseling assistants, and serve patients both on the inpatient and outpatient side of the hospital. I also have a focus in quality improvement and process improvement. So I spend a significant amount of my time looking at how we care for patients and their families and how to institute better processes. How do we better triage patients, make sure they get to the right physicians? How do we help work patients through getting their labs done, getting all of the support documentation? And if there are times where those systems don't work well, how do we look at that and how do we do better? And so those are some of my areas of focus. The first speaker that we're going to have talk to you is Dr. Jackie Omorodorong from Boston Children's Hospital.
Thanks, Amy. So here, let me just share my slides and I'll get started. So as Amy mentioned, my name is Jackie Omorodorong, and I'm one of the clinical geneticists in the Division of Genetics and Genomics at Boston Children's Hospital, and also on faculty at Harvard Medical School. And I'm really excited to be chatting with you all today about exactly what I do as a clinical geneticist. And so to get started, just a little bit of information about myself and how I got onto this career path. So I actually grew up in a small town called Newmarket, Ontario, which is just north of Toronto up in Canada. And so lived there all my life until I went to college. And so I went to college at McMaster University, where I majored in molecular biology and genetics. And so I went into college kind of knowing that I wanted to be a doctor and I wanted to go into medicine, but didn't know exactly what I wanted to do. And at the same time, I had a real passion for genetics. And so it wasn't until my third year in college that I actually took a medical genetics class and fell in love with it. And so I'll tell you guys a little bit about what we do in medical genetics, but I absolutely loved it. And so after that, I went to medical school in Washington, D.C. at George Washington University, and then afterwards moved to Boston. And so here I did a combined program in which I was able to do my pediatrics residency through the Boston Combined Residency Program and my genetics fellowship through the Harvard Medical School Genetics Training Program. And so I actually just recently finished this past June. And so I've been an attending physician or supervising doctor for a couple of months now. And so this is what a typical week for me looks like. It's really broken down into 3 big parts. And so Mondays and Tuesdays, I spend all day in clinic, and really I'm seeing different types of patients. Wednesday and Thursday is then kind of post-clinic work and pre-clinic prep that I'll talk about. And then finally, I actually spend a day of the week working on research too. And so starting with my clinic days, what are my clinic days like? So we all have tens of thousands of genes in our body. And when there's a mistake or a problem in some of those genes, there are certain type of features or medical problems that you may see in a patient. And so certain features or findings that we see that often result in a referral to genetics would be something such as multiple congenital anomalies. And so congenital anomalies are differences that people may have at birth. And so some examples would be a cleft lip or palate, an extra finger or toe, a hole in the heart, a problem with the kidney. And so when we see multiple findings like this in one patient, that makes us think, could there be a problem with their genes, and that they come in to see us? Other things that may result in a referral to genetics would be a neurodevelopmental difference. So if somebody has developmental delay where they don't start speaking or walking on time, or maybe not at all, they might come to see us. Or if they have a neurodevelopmental difference such as intellectual disability or autism spectrum disorder, they may come to our clinic as well for us to see, is there a change in their genes? And then finally, another common one that I see is if there's a concerning family history. So we might see a patient where, they have cousins, aunts, uncles, grandparents that all have a condition. And so now I'm seeing a patient to see, do they also have this condition too that seems to be running in their family? And so we see a mix, and this is kind of a list of the bigger buckets. And for me, if I'm being completely honest, for me, clinic days are the best days. So I really enjoy working with patients for a number of reasons. And so I think one, as a pediatric geneticist, I'm working with younger patients who are often coming to clinic with their parents. And so I love getting to work with the kids and their parents. I'm learning about their history and the family history as well to really be able to figure out what's going on. But also, just given the nature of our genes, I get to work with families. So it's not uncommon at all for us to see siblings who have the same diagnosis, or to maybe follow a set of cousins that have the same diagnosis. And so it's really nice that you can get to know the entire family a bit better and work with multiple different members of the same family. And then another piece that I really enjoy, and that I did a lot of today in clinic actually, is that there's a large teaching component in genetics. So I think in medicine as a whole, we kind of owe it to our patients to be able to teach them and explain to them what exactly we're doing and why they're seeing us. But I think in genetics, this is an even bigger component because people aren't as familiar with their genes. It's not something we talk about as often as we do like the brain or the heart. And so there's often a teaching component to our clinic visits, which I really enjoy. And so that's kind of what my Mondays and Tuesdays look like. And then my Wednesdays and Thursdays, as I mentioned, are really kind of wrapping up what I saw in clinic the prior 2 days and then getting ready for clinic the next week. And so when I think about genetics, I often think of this question that my best friend gave to me when I was in medical school. And I told her, I've decided I'm definitely going to do medical genetics. And so I explained it to her. Her response was, so you're pretty much going to be a medical detective then. And I think that that's an accurate point because, as I mentioned, we have these patients come into us that have different either physical differences or developmental differences. And it's up to us to kind of figure out what exactly is going on. In our tens of thousands of genes, where do we think the problem is? And so in order to kind of figure it out, we do a lot of review of the literature. So reading about patients that have presented similarly in the past, reading about genes that may lead to different problems that we've seen in our patient, and just learning about what kind of emerging therapies might be out there. And so with this, we also will kind of decide what kind of testing do we want to offer the patient. And so as you may remember from science class, we have a bunch of cells in our bodies, and in our cells are the nucleus, which have our chromosomes. And then within our chromosomes are all of our DNA that compose our genes. And so from a genetic standpoint, we're able to offer testing at different levels. We can think about, do we think the problem is at the level of the gene, or do we think the problem is at the level of the chromosome, for example? And it's on us to decide where do we think it is and what kind of testing would be most appropriate. So I've really enjoyed that piece too, because you're always kind of learning as you see new patients and see new presentations that maybe you haven't seen before. And then finally, For me, my Fridays are composed primarily of research. And so in genetics, you have the opportunity to be more clinic focused and work very closely with patients, or to be more research focused and kind of spend the majority of your time on that. And so for myself, I'm primarily clinical with a little bit of research, but every kind of combination exists, from people that are more clinical to people that are more research, and then some people that are half and half. And so for me, I'm primarily clinical, but I do about 20% research. And in genetics, you have the opportunity to do research really across a broad spectrum of diagnoses, groups of disorders, different genes, but also just areas of interest. And so for me, a lot of my research is focused on the ethical, legal, and social implications of rare disease treatment. And in particular, I look at health disparities and health inequities in the rare disease population. And so I think getting to actually work on research and contribute to the literature that exists has been really nice. And it's been a lot of fun, because questions that you come up with yourself, you're able to work with colleagues to kind of answer the question and contribute to the literature. And so this is a paper that I published with a few colleagues across the country as a fellow, just looking at potential racial biases in kids with genetic conditions. And then here, this is a poster that I presented at one of our medical conferences last year, just looking at parental attitudes about genomic screening. And so being able to attend academic conferences is a lot of fun, because you get to meet and kind of network with a lot of medical geneticists, biochemical geneticists, genetic counselors, and really just a bunch of other people that are interested in the same area that you are. And so it's always a lot of fun getting to be a part of that. So I'm really happy that I get to do some research. And so for me, I just wanted to wrap up with, why exactly do I love genetics? And so some of the things I've already touched on are the fact that we get to work so closely with patients and their families, the fact that we're always learning because there are so many genes. But a few of the other things, though, is that I really like that with genetics, I didn't have to pick one single organ system to focus on. I really got to think about the entire person and think about the entire body. For me, I'm a pediatric geneticist, as I mentioned, so I really like the fact that I have the opportunity to work with kids in this field. But through genetics training, we're actually trained to work with people of all ages. So I like the fact that, if down the line I decide I wanted to work with adults, I'd have the opportunity to do that. If I decided I wanted to work primarily with pregnant patients, I could do that. Or if I wanted to see a mix, I could do that as well, because genetics impacts all of those different patient populations. I like the fact that in genetics, we have the option to conduct research if we'd like, often very closely related to what we're seeing in clinic. But again, it's an option and not something that everybody has to do. And then finally, I like the fact that I can be a clinical geneticist in an academic environment, And so I really like the fact that being in an academic environment, I get to work with college students, medical students, residents, fellows, and kind of all work together and collaborate on different projects, but also in medical education throughout. And so for me, this is a little bit of what I do, and a little bit of reason why I love this field. But thank you guys for listening. And so I've included my email here in case there are any questions that we're not able to get to during the Q&A, or if you guys have any questions about a career in genetics at all, I'm happy to chat.
Thank you so much. And we will definitely be able to get everyone's contact information if someone has further questions outside of this. I think you will find, as you're already seeing with Dr. Omororan, we love to talk about our field, and so are always eager to talk to people who are interested. We're going to switch gears a little bit now, and we're going to have a wonderful opportunity to hear from Dr. Leck and the work that he does.
Well, thanks for the introduction. And while I'm sharing the screen, I didn't prepare a slide for this, but I'll talk about how I got into genetics. So it's a very unique story. You may not know, because I'm just sitting here, that I have a rare genetic disease. And so I got into genetics and I left a career in IT. I worked in IBM because I wanted to contribute to the gene discovery efforts, and also and the downstream efforts in terms of developing therapies also. And that's how I got into genetics. So it's a quite long and windy story, but that's how I got into it. So I thought to present to you a day in the life of being a professor in genetics. So I'm going to talk about a research project that runs out of Yale and is one of the flagship projects supported by the NIH, then NHLBI. So that you get a taste of genetics research, and this is what I do on a full-time basis at Yale School of Medicine. So So the project's on congenital heart disease, and congenital heart disease has an incidence in 1 in 100 live births. So it's quite a common condition seen in pediatrics. And it's really good for genetics because it's what we call a common rare condition. So we don't have issues in actually recruiting patients for genetic studies. So in the US, it's estimated to occur in 40,000 infants annually, and surgical procedures and medical interventions are required during infancy in about a third of the patients. 90% fortunately now survive to adulthood, and now there's an increased CHD prevalence amongst the population. So this is how the etiology looks like, and the common causes of congenital heart disease, you know, and the breakdown in terms of genetics, environmental causes, and the different genetic mutations cause it. So I think Jacqueline did a fantastic job of introducing DNA mutations, genetic variants, and things like that. So I won't get into little details, but one of the challenges with all rare disease genetics is the unknown, and typically the unknown for a lot of rare genetic diseases, about 50%. So 50% don't have a genetic diagnosis. And this is where the area of research is, what makes up that gray area. So, and this is the goal of the Pediatric Cardiac Genomics Consortia, to actually make that impact. So it has been going on for quite some time yet. It was established in 2010. There are multiple centers across the United States, including Yale. Harvard Medical School is one of the sites. So we collaborate with the Simon Lab, based in BWH, who's been working on this project since the start also. We perform whole exome and genome sequencing of patients, so either sequencing all the coding regions of genes, of the 20,000 or so genes, as Jacqueline had mentioned, to look for variations that are associated with congenital heart disease. We also, and this is the best thing about the cohort, we have a comprehensive curation of phenotype data. So where medical staff have actually curated that data and made that available for analysis also. So at the moment, we're at 11,000 probands and 4,000 trios. So what I mean by probands are the affected, and the trios is when we sequence mum, dad, and also the child to look at variants that have been transmitted or new variants. So this is how the cohort has grown in terms of size, in terms of recruitment. So we are very grateful research coordinators, genetic counselors, and all those involved in this massive recruitment effort. So this today is the largest cohort of congenital heart disease that I'm aware of in the world. So it's really great for genetic discovery, where we have collected DNA, and that's the breakdown of how many trios we have now in 2023. So we've increased it a lot since the last iteration. So the problem is, with our current cohort, we're trying to find the cause for the remaining 56%. So that chunk of the pie chart. And why is this the case? And I'm not going to go into the technical details, but these are some of the innovations we're going to bring. So the hypothesis is, by increasing the cohort size, if it's a numbers game, it should actually lead to more discoveries. And also, we are also improving different methods that we go about identifying the genes. As Jacqueline was saying, being genetic detectives and trying to find the genes and the variants that underlie congenital heart disease. The other thing we've shown is a projection of where we're at today. And so we're at eight genes that have been statistically significantly associated with congenital heart disease, and what we expect as we increase this cohort. So yes, it doesn't sound amazing. We're going to go from 8 to 14 genes that we project, but this is still a big advance in terms of things that are causing congenital heart disease, genetic causes. So the analytical workflow of what we do here is that after the sample is collected, we perform sequencing. So this is using next generation sequencing. When we get the raw sequencing data, we align it to the human genome. Then we look for genetic variants that are different when you compare a patient's genome with the human genome. We then use methods to analyze it, do quality control, look at the variants and annotate them. So the variants can be annotated by whether they say impact the amino acid sequence, so they're called missense variants, or if they create loss of function. So these are mutations that may impact splicing, create a nonsense mutation, etc. And then we perform some analysis. So what I'm going to touch upon is what analysis looks like when we're looking at de novo variants. So what we found in the over decade of studying this cohort is one of the largest contributors to congenital heart disease is actually de novo variants. Well, when we look at it in this way of analyzing data, so de novo variants are variants that do not exist in the parents, but are present in the next generation of the child. So the thought is the mutation either occurs in the egg or the sperm, and that's why it's not actually found in the germline, so in any other cells in the parents, but is actually found in the child. This is what we call a de novo variant. So on average, if you do the right data controls and filtering, we expect somewhere between 0 to 3 de novo variants in coding regions of the genome. So things that impact the protein sequence itself of genes. So, and from this, we also then provide functional annotation on the functional effects of those de novo variants. So we can then ask questions in terms of statistical tests, in terms of a single variant test. So whether a variant is associated with phenotype is typically performed in a genome-wide association study. And this is applicable when the variants are common, but it's hard for rare variants. So we believe that a lot of the variants underlying congenital heart disease is actually rare variants. So instead, what we do is what we call a burden test or a gene burden test. We take a gene and ask the question of how many damaging de novo variants do we see in our cohort of congenital heart disease compared to an expectation or a control cohort, and ask the question, is this statistically significant? I'm not going to go on the mathematics of it, but that's pretty much the question we're trying to ask in here. So in other words, do de novo variants mutations in a particular gene occur more often than expected or more than in a control cohort? And so this is typically how the data looks like. So I'll step you through this. Each one of these dots represents a gene in the human genome. So there's somewhere around about 20 to 25,000 genes, depending on what you consider as a gene and annotations you'll use. So on the x-axis, we have the expected, and on the y-axis, we have the observed. So what we see in the cohort, expected could either be the control cohort or what a particular model, usually a mutation model, predicts. And normally what you expect is a lot of these genes colored in black should lie on the red line. So that's saying what we expect is actually what we observe, and therefore it should not be associated with congenital heart